enzyme-linked immunosorbent assay, also called as ELISA. ELISA is an enzyme immunoassay commonly used for detection and quantitation of antibodies, antigens, proteins, and glycoproteins in biological samples. It may sometimes refer to as solid phase immunosorbent assay or simply an enzyme immunoassay. It is a test with high sensitivity and specificity. It is a solid phase type of immunoassay that involves specific antigen-antibody interaction in vitro. It was first described by Eva Ingwall and Peter Perlman in 1971. ELISAs are typically performed in 96-well polystyrene plate, also called as microwell plates. Various components of ELISA are antibody, that is IgG fraction of serum, enzyme HORS, that is peroxidase, a glycoprotein with 4 lysine residue, substrate TMB, that is 3,3', 5,5' tetramethylbenzidine. The enzyme acts as a catalyst to oxidize substrate in the presence of hydrogen peroxide to produce a blue color. In general, the principle of ELISA depends on having the reactants of the ELISA immobilized to the microplate surface, making it effective to separate bound from unbound materials. This ability to wash away non-specifically bound materials make the ELISA a powerful tool for measuring specific analytes in a sample. When an enzyme labeled molecule is added to the well containing specifically bound antigen or antibody to receptive antibody or antigen respectively, the addition of a substrate gives colored reaction. This colored reaction can be measured by ELISA reader to detect and quantify the test analyte. ELISA begins with a coating step in which the first layer consisting of the target antigen or antibody is immobilized onto a 96-well polystyrene plate. This is followed by a blocking step so that all the unbound sites are coated with a blocking agent. Following a series of washes, the plate is incubated with the enzyme conjugated antibody. Another series of washes removes all unbound antibody. A substrate is then added, producing a colorimetric signal. Finally, the plate is read. The coating step is not required if using a kit with a plate that is pre-coated with antibody. Also, during washing, excess liquid is essential to get removed to prevent dilution of the solutions added in the next step. There are 4 basic ELISA formats. They are direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Direct ELISA. It uses the method of directly labeling the primary antibody itself. Microwell plates are coated with a sample containing the target antigen. Enzyme labeled known antibody and substrate is added. The binding of the labeled antibody gives color, which is quantitated by colorimetry. Various advantages are: chances of cross-reactivity of secondary antibody is 0. Protocol is less cumbersome and short, as only one antibody is used. Methodology uses less amount of reagents. Disadvantages are: immunoreactivity of primary antibody may be affected due to its labeling. Potential for high background may affect the sensitivity of the test. It is less specific as single antibody is used. No flexibility in choice of primary antibody label, and also there is no signal amplification. Indirect ELISA. This assay detects the presence of antibodies in a sample. It utilizes an unlabeled primary antibody in conjunction with labeled secondary antibody. The secondary antibody is usually an anti-species antibody and is often polyclonal. After the addition of substrate, developed color is measured by using ELISA reader. The antigen is coated to the microbial plate is detected in two stages. First, unlabeled primary antibody specific for the immobilized antigen is allowed to bind to it. Next, an enzyme labeled secondary antibody is allowed to bind to the primary antibody. Lastly, after the addition of substrate, the developed color is measured by using ELISA reader, either directly or by stopping the reaction using suitable stop solution. The rate of color development is proportional to the amount of antigen. Advantages are signal amplification is there and so increase in sensitivity of the assay, as each primary antibody contains several epitopes that can be bound by labeled secondary antibody. It is more flexible as the same secondary antibody may be used for several primary antibodies. Wide variety of labeled secondary antibodies are available commercially. Immunoreactivity of the primary antibody is not affected as it is not labeled. Disadvantages are cross-reactivity with secondary antibody results in non-specific signal. It is time-consuming as compared to direct ELISA due to long protocol. Sandwich ELISA. Sandwich ELISAs typically require the use of meshed antibody pair to determine antigen concentration in sample. Each antibody pair, that is capture antibody and detection antibody, is specific for different non-overlapping epitopes of the same antigen molecule. Capture antibody is the first antibody that is coated on the plate to immobilize the antigen, followed by the addition of sample solution to the wells. Detection antibody is the second antibody that is added to bind the antigen captured by the captured antibody and enable subsequent measurement of the concentration of the analyte. If the detection antibody is conjugated to an enzyme, then the assay is called direct sandwich assay, ELISA. If the detection antibody is unlabeled, then the secondary detection antibody will be needed, resulting in indirect sandwich ELISA. Direct sandwich ELISA uses a conjugated detection antibody. Following incubation with the antibody-antigen complex immobilized on the plate well, signal detection is performed. Indirect sandwich ELISA, it uses unconjugated antibody bound to the antibody-antigen complex on the well. Following removal of unbound antibodies, the remaining antigen-bound antibodies are targeted by the conjugated secondary detection antibody, and the signal detection is performed. Advantages: it is more sensitive than the indirect ELISA. High specificity as protocol involves pair of antibodies recognizing different epitopes on the same antigen. Suitable for detecting antigen content in a complex sample effectively. More flexible as both direct or indirect method can be employed. The antibody coated plate can be stored and used whenever needed. Disadvantages are finding two compatible antibodies against different epitopes on the same target can be challenging to develop. The antigen should have at least two specific sites. Competitive ELISA. It is used to detect antibodies in unknown samples. Basic principle is the competition between two different antibodies present in the sample and competitor antibody mostly conjugated to bind on the same antigen. Advantage is that this type of assay is suitable to quantitate small molecules, and disadvantages of the assay is less specific since only one antibody is used, and the assay requires a conjugated antigen. Applications of ELISA. Number 1. Screening of donated blood for evidence of viral contamination by HIV-1 and HIV-2, hepatitis C virus and hepatitis B virus. Number 2 is measuring hormonal levels like that of HCG, LH, TSH, T3 and T4. Number 3, for measuring and monitoring the humoral immune response in the host against the disease. Number 4, a number of different commercial ELISA kits are available for detecting a variety of disease types in human and animals. Number 5, detecting allergens in the food and house dust. Number 6 is measuring toxins in contaminated food. Number 7 is detecting illicit drug, example cocaine, opiates. Number 8, this technique is of great help to avoid possible fraudulent substitutions in food industries. Number 9, it serves as a great candidate for vaccine development by screening the presence of desired antibody levels developed in the experimental host tested against test antigen. And number 10, measuring rheumatoid factor and other antibodies in autoimmune diseases like lupus. Don't forget to subscribe to the channel and hit the notification bell to remain updated for more such videos. Thank you for watching.